This is one of several related presentations by New Liberty Proteomics on the drug Nelfinavir. Here we look at its surprising behavior as a proteolysis promoter. Nelfinavir, trade name Viricept, was launched 14 years ago, and that launch was the most successful biotech launch in history at that time. Nelfinavir binds to and inhibits the activity of HIV protease, thereby inhibiting replication of the virus. Through a likely connection to the drug's side effects, Nelfinavir is being tested as a treatment for a variety of cancers. Let's look briefly at New Liberty's proprietary methods and the bipolar nature of Nelfinavir. Proteases digest proteins into smaller pieces, and smaller objects rotate more rapidly. This trace shows full angular immobility of a single labeled amino acid in serum albumin. Following a 7-minute digestion by trypsin, a second pattern begins to emerge, representing a much more mobile subpopulation. This is direct physical observation of whole protein digestion. As proteolysis proceeds, the mobile population increases in concentration, with a corresponding decrease in immobile, undigested substrate. Let's look at the actual data demonstrating the substrate-dependent behavior of Nelfinavir. In black is the spectrum of hemoglobin following a 40-minute digestion by trypsin. If Nelfinavir is present, we see a 20% decline in the mobile population. Therefore, if hemoglobin is the substrate, the drug acts as a protease inhibitor. Here we change only the substrate to serum albumin. The presence of Nelfinavir produces an increase in mobile population, demonstrating its apparent ability as a promoter of proteolysis. Let's summarize and examine this most peculiar behavior. NLP methods uniquely monitor digestion of whole protein substrates in real time, both requirements for these surprising observations. As we've seen, Nelfinavir inhibits the digestion of hemoglobin by trypsin. Inhibition by the drug has been seen for proteases from multiple sources and classes, as is shown in our companion videos. In contrast, Nelfinavir promotes the digestion of serum albumin, again for the same multiplicity of proteases. These data have several implications. There is an apparent competition for the drug between substrate and protease. Nelfinavir binds to both proteins. The drug-albumin interaction is not passive. Rather, Nelfinavir renders the substrate labile to proteolysis. Finally, the drug is seen to be lipophilic, as albumin is known as a general binder of lipophilic materials. In this next to last slide, both observations and questions arise. It is widely known that many drugs interact with albumin and with membranes. Are these interactions passive, as is often assumed? Our data argue otherwise. Should we exploit testing, such as represented here, as a routine part of side effect discovery? Are there implications here for Nelfinavir's action as a possible cancer treatment? For example, it's known that the drug indirectly inhibits the protein AKT in cancer cells. It's also known that AKT binds phospholipids. Does our work present a possible mechanism for AKT inhibition by Nelfinavir? It's certainly worth a look. We take up this question in Nelfinavir, the Cancer Treatment, Part 2. Until then, we're New Liberty Proteomics, and thanks for dropping by.